It's a pleasure to be here. So my lab in IIT Bombay is a microfluidics and biological physics lab. And we work on different aspects of microfluidics. So the current projects involve isolation of extracellular vesicles, droplet microfluidics for a bunch of applications. We also look at bacterial motility, how it changes under microfluidic confinement. And we look at shape-based detection of sickle cell disease. But today, what we are going to talk about is how we use microfluidics to measure stiffness, the Young's modulus of single red blood cells. So, right. So let me just give a very quick recap about blood cells. Red blood cells, we know they are healthy. Red blood cells are biconcave in shape and they are very, very deformable. And it's the deformability that we are actually going to talk about today. Okay. So if you look at normal RBC, its shear modulus ranges somewhere between 2 to 10 micronewton per meter. But if a red blood cell gets infected by the malarial parasite, Plasmodium falciparum, then at different stages of development of the parasite, the stiffness of the blood cell goes up. So you can see that if using optical tweezer, if we measure the stiffness, we can see quite a distinct jump in stiffness as the parasite develops, okay? And these are the images that people have measured earlier, taken earlier when they tried to stretch the red blood cell with two beads connected on two sides using an optical tweezer. Similarly, sickle cell disease is a hereditary disorder where when people inherit the sickle hemoglobin, under low oxygen conditions, this sickle hemoglobin actually polymerizes. And it is that which leads to stiffening of the red blood cell, which also leads to these characteristic shapes, which are called the sickle shapes. Okay? And again, if you measure the stiffness of individual sickle RBCs, you see in sickle disease, the stiffness changes quite a bit. It's more than 150 micronewton per meter. And sickle trait is a condition where an individual has both normal hemoglobin as well as sickle hemoglobin. So although it changes, the shear modulus increases, but still not to the same extent as having sickle cell disease. Okay. So we were looking at different microfluidic techniques in the literature to see how we can measure stiffness of red blood cells. And there are quite a lot of papers, but many times one of the things that happens is that people either use very, very fast cameras to measure the shape changes as a red blood cell moves through a constriction, or sometimes it's another surrogate measure like the transit time, etc., that accounts for the deformability of the red blood cells. At this point of time, we also realize that when we look at the conventional techniques, we find that there are different techniques, right? There's optical tweezers, there's micropipette aspiration, AFM, and so on. And each technique actually measures something very different. So some of them would measure the shear modulus, which is perhaps the property of the cell membrane. Some other techniques would measure Young's modulus, which is the property of the entire cell. And it's very difficult to compare between these two properties. Plus, the throughput of such measurements is quite low, as it's quite expensive as well. And so this is where microfluidics could really play a big role. So at this point of time, we came across a paper. This was by a collaborative group in Sweden. So they did numerical simulations of capsules, which are basically spherical liquid-filled object bound by a membrane, passing through a microfluidic device. So there's a straight channel that opens into a funnel with an obstacle at the mouth of the funnel. Now, the capsules that are actually soft, the blue ones, if they come across this line and approach the pillar, when they pass the pillar, they actually pass quite undeflected. So their deflection's very, very little. However, if the capsule happens to be stiffer, then they deflect by a larger angle. So this was a numerical study, and this observation was quite interesting. And a few years down the line, actually, I think a few years down the line, we saw this another group in the University of Compiègne. They actually did this similar experiment, but this time they used lycopodium particles and vesicles and so on. And they also verified that what this group had predicted, it actually works. So objects with different deformability, they actually leave this funnel at larger angles. And that led us to think, can we actually extend this to cells? And the RBCs are the simplest of the cells in that they don't have a nucleus. So we thought, let's start testing this device with red blood cells. So this is what our microfluidic device looks like. So it has sort of three parts. There's an initial flow focusing part, then there is a deformability measurement region, and finally there's a sorting area. So over here, we pass blood. It's quite diluted, about 1,000 times or so. And then over here, we pass the buffer. So the RBCs, when they approach this pillar, they are actually focused and they approach along a single line. Now, if you see here, you will see the distance between the side of the pillar and the channel wall is about 10 micron on each side. So the RBCs are not actually being squeezed by the constriction, but they will actually be squeezed by the flow when they go here. So this is the area where we actually take images and we try to see how the RBCs actually, at what point they leave this region. And then once they leave this region, these are a bunch of outlets. So you can actually sort RBC fractions by deformability in these outlets. So these would be the softer ones, and the ones at the edge would be the stiffer ones. And the entire microfluidic device height is 5 micron to ensure that the RBCs do not flip on their side when they go here, but they all remain on their faces. Now, this project was started by my first PhD student, Ninaz, towards the end of his PhD, and it has been established completely by the second PhD student, Savita. So the hypothesis is that if we have RBCs passing through this device, the softer RBCs would follow this green line and the stiffer RBCs would follow something like this, the blue line over here. And all these experiments, since we are simply monitoring the track and not the shape change, we can actually use much slower camera. So by the way, this is a microscope that's developed in the lab by another different student. This is the microscope where we do all our microfluidic experiments. It runs on an iPad camera with just its 30 frames per second images, and that helps us do these things. Now, coming back to this, this is the video that you can actually see the tracks of the RBCs passing here. So what happens is that as an RBC comes through here, it's squeezed, and its center of mass, it actually shifts. If it's really soft, then its center of mass remains actually closer to the pillar. But if it's not being squeezed much because it's already stiffened, then your center of mass remains on a different streamline. And because it's laminar flow, at this point, whatever streamline it's lying on, it will also exit the funnel at that same streamline. So if I can simply measure where they're coming out, I can actually figure out which one is stiff and which one is deformable. Okay. So now the point is that if the RBCs are coming out this way, I have to have a way to label the RBC positions. Okay. So to do that, so this is our experiment. And then what Savita does is that she does a console simulation of the fluid flow in this device without any RBCs. So that gives her a set of streamlines. So she numbers these streamlines, the center one being 0 and 50 to each side, 0 to 50, okay? And then what she does is that she does some image processing to superpose the experimental frames, all of them on this simulation frame. That way we get a coordinate system. So if we know where the RBCs, we actually label its position using a streamline, okay? And then what she does is she actually plots this distribution. So there are these, the streamline number is on the x-axis, and this is the PDF. And as expected, most in a healthy population, most of the RBCs should be soft, and therefore they lie along streamlines, which are quite small in numbers, so somewhere around this area. And you can see that we can sort of measure about 25,000 RBCs using this technique. Now the challenge here is that now that we know their position, we have to assign an elastic constant to this, right? And the simulation that was reported, which was the inspiration for our work, they assumed spherical capsules, whereas RBCs were biconcave with different material inside and outside with different viscosities. There's constant exchange of material. So all the simulation that we tried to do to model the RBC at this point had completely failed. So at this point, we thought that let's look at a different way of finding out an elastic modulus. So what we did, ideally what we would like to do is that we would like to collect our RBC from the outlet of our microfluidic device, take it out, measure its elastic modulus using a different technique and generate a calibration curve. But that's practically not possible. It's logistically quite impossible. So what we did is we took the same healthy individuals' RBCs and then we performed AFM on them. And you can see that the number of measurements or the number of RBCs that we could measure is about 700. It's still a large number for AFM, but not a large enough number compared to the microfluidic experiment. And the AFM experiments were done with my collaborator Shamik and his PhD student Tanushri. So now what we have is we have one distribution from AFM and another distribution from microfluidics, which gives you the position of the RBCs. So how to relate to that? And this is what our calibration curve does. So what we do is we first have our experiment from which by combining the experiment with COMSOL, we get this distribution. We also do AFM and we get a second distribution. And this is where my collaborator Dhruva, originally who published the simulation paper, we worked with him to develop a calibration curve. So what he did was a data-driven calibration. So we asked ourselves that if these two are the distributions, if I assume the Young's modulus is a function of the streamline, and then we want to know what is this mathematical functional form that would actually overlap these two distributions on each other. In other words, the sort of minimize the Jensen-Shannon divergence between these two. And in doing this, this is basically we assume a polynomial and then we run this and we find that a fourth order polynomial is actually a calibration curve that we get. Okay? So this is the equation of the calibration curve. So you have the streamline on the x-axis and you have the Young's modulus on the y-axis. This blue curve is the calibration curve. Now, this calibration needs to be done only once, and after that you can use it to validate with any unknown populations with some constraints that your device remains the same, your flow rate remains the same, and the shape of the RBCs or the size of the RBCs does not change. So after getting this calibration curve, then we went on to validate this. So first what we did was we artificially stiffened healthy RBCs by treating them with the chemical glutaraldehyde at different concentrations. And you can see that as we keep increasing the glutaraldehyde, the mean stiffness of the distribution also keeps changing. Okay, so this is as we expect. Now, so this was something that we did in the lab, and then we wanted to see that how does it work for, say, a disease condition. Now, here there's something that we have to keep in mind, which is that is the size of the RBC changing when we are treating it with glutaraldehyde? And we did this. These are the two distributions. And we find that the mean of these two are really not very different. So what we are seeing is actually because of deformability change and not because of size change. Then we took these two RBCs, soft and stiff, mixed them in 50% ratio, and then we passed them through this device again. And as expected, this green plot, it gives us a bimodal distribution. So the blue plot is for the healthy RBC and the orange one is for the stiff RBCs, and the green one contains a bit of both. So this tells us that we can actually use it as a deformability-based cell sorting device. Then we started looking at what happens to red blood cells when they are stored. We store blood for up to 42 days for blood transfusion. And so we took blood from a healthy individual and we stored them this 3% at 4 degrees. And every third day we took it out and we measured the stiffness. And as you see here, you see that up to a week, the stiffness doesn't change much. But beyond that, the stiffness starts changing. And at about 2 weeks, it's quite different. Okay, so this is something that's actually useful for us to know because it's quite relevant for blood transfusion where we transfuse blood up to 42 days. And if the blood is already stiff, the RBCs are already stiff, then they would degrade and you would need to do transfusion immediately. Okay, so that was the second case study. The third case study was with malaria-infected RBCs. So my colleague Swati Patakar, who studies malaria here, and a student Chaminder and my student Priyanka, we started working together. So what we did is we took 4% the malaria in blood with 4% parasitemia. That means only 4% of the RBCs have parasite in them. But very strangely, we found that, yes, we do get two different peaks, which is what we would expect. But given it's only 4% parasitemia, I would have expected these peak to be like a really small blip. But on the other hand, it looks like there's quite a lot of stiff RBCs in that population, although they might not have directly parasite inside them. Okay, and this is something that we are still investigating. It's unpublished data. This is what we have initially. Okay, and we also work with sickle cell, but that's I'm not going to go into it today because that's a longer story for another day. So I'll stop here by acknowledging my students. Savita Kumari is the one who's behind this entire project. She's kind of really formulated it and brought it to the state that it is now today. Ninad and the others, they have also worked here in this technology. My collaborators Dhrubo, Shamik, and Swati are the ones who I've worked with, and the funding agencies, all of them are acknowledged, particularly IIT Bombay, Gates Foundation, Vadvani, etc., who have really supported all the different blood-related projects here.